Hello, everyone. My name is Victoria Tuttle. I just started graduate school and I will be pursuing a master's in animal science with an emphasis on physiology. I've been working for Dr. Fraley for over a year now, and this presentation is an overview of what I've been doing the past year as an undergraduate student. And today I will be reinvestigating the adrenal cortex function as it may pertain to the stress response in adult pekin ducks. For the topics I'll be covering today, I will start off by giving a brief background and talk about any relevant information that will help you further understand my research. I will then go into my hypothesis and discuss the materials and methods involved. I will then interpret my results and finally discuss any potential future studies that I may be interested in pursuing further. So to start off with the background on the HPA axis of mammals and birds, HPA stands for hypothalamic pituitary adrenal axis. So it has interactions between three main components. There is a bit of variability in which glucocorticoid is dominant in animals. For example, most mammals and fish secrete cortisol, while rats and mice secrete corticosterone, and dolphins are actually ones to secrete both. In birds, it was believed that corticosterone was the predominant glucocorticoid. However, there were many reports of changes in cortisol levels in the stress response in chickens, and there were numerous similar findings in various wild bird species, which I'll get into in the next few slides. But the main difference between the two axes is that the avian HPA actually utilizes two neurohormones, which are CRH and AVT. CRH is corticotropin releasing hormone, and AVT is arginine basotosin. So this is the mammalian HPA axis. CRH is located in the paraventricular nucleus of the hypothalamus. When we are stressed, the hypothalamus releases the CRH, which then signals the anterior pituitary to secrete ACTH, which then travels down to the adrenal glands, where it stimulates the release of corticosterone and or cortisol. For the avian HPA axis, CRH is located in the thalamus and AVT is in the hypothalamus, and both stimulate the anterior pituitary to secrete ACTH, which then goes to the adrenal cortex to secrete stress hormones. So as of now, we don't really understand the differential functions in CRH and AVT in birds, so we want to figure out more about, do certain stressors initiate the release of CRH versus AVT? So both primary glucocorticoids are synthesized from this common pathway that begins with cholesterol and pregnenolone. However, after synthesis to pregnenolone, the pathways diverge to produce either cortisol or corticosterone. It's also important to note that the synthesis of one glucocorticoid does not depend on the synthesis of the other. So there have been many past studies done with ACTH and stress in birds. We know in poultry that there are some stressors, such as transportation stress, that have mixed results on corticosterone or HLR levels. So HLR stands for heterophil to lymphocyte ratio, and this is important to know because it is an indirect measure of stress. So in response to stressful situations, glucocorticoids increase HLR because there is an increase in blood heterophils due to increased migration from the lymph nodes, and there is a decrease in lymphocytes due to marginalization. There were also mixed reports from various studies about the stressor's effects on HLR. Previous studies have injected ACTH into birds and have seen an increase in corticosterone levels and mixed effects on cortisol. There was also another study done where ACTH was injected into ducks, again showing an increase in corticosterone, but cortisol was not measured. Other studies have shown that chronic stress reduces the size of the bursa fabricius, but this is unrelated to corticosterone, but it's still hypothesized that there is an unknown factor associated with stress in birds. So we hypothesized that cortisol is differentially released in response to ACTH compared to corticosterone in ducks. Overall, with this project, I wanted to be able to show that both glucocorticoids have a physiological function in adult drakes and hens. So for my methods, I utilized adult breeder Pekin ducks from Maple Leaf Farms. They were between 35 to 40 weeks of age, and hens and drakes for approximately 4 to 4.5 kilograms respectively. I also utilized an 18:6 light cycle with lights on at 0300 hours with food access for 8 hours per day. The ducks were injected IM with cosentropin, which is an artificial ACTH, and saline was used as the control. The treatment also had 3 dosages, which were low, medium, and high. We collected blood at 4 different time points with injections beginning at 0730 hours. And this blood collection was used to measure corticosterone, cortisol, and HLR. And HLR were then determined in an independent lab by avian pathologists who were unaware of the treatment groups. So the ELISA kits that I used were from Cayman Chemicals, and these were previously verified by Dr. Fraley and his lab. I was not present at the time he was going through this verification process, so I will just briefly summarize what he did. Activated charcoal was used to strip the duck serum of any steroids, and he spiked back known concentrations for the standard curve in the serum. And this was done to make sure that there was no measurable cross-reactivity between the kits to the respective glucocorticoids. And this was done to make sure that the cortisol ELISA kits only gave values for cortisol and vice versa. Mass spectrometry was also used for this verification process. Data was analyzed by 3-way repeated ANOVA with p-values below 0.05 considered significant. And cortisol and corticosterone were not analyzed together because since they are 2 separate hormones, they require different kits and they have different incubation times. So all the analyses were individual. So now getting into my results, this is corticosterone. The 2 graphs are separated by sex to make it easier to visualize, but also to note the sex differences. So there is a dose response effect, and there is also a significant sex by dose by time interaction with all responses within physiologic ranges. So the lowest dose, represented by the red line, had no effect on the hen secretion of corticosterone and only in the drake at the 1st hour. For cortisol, there was another dose response effect, and you can clearly see the different patterns of release between the sexes. There was also a significant sex by dose by time interaction, and the lowest dose had no effect on cortisol secretions in either sex. Finally, for HLR, there is no dose response effect, but there is a significant sex by time interaction. So even at the lowest dose of ACTH, which is also represented by the red line, that had no effect on the glucocorticoid secretion in either sex, still elicited an increase in HLR. So for my results, ACTH stimulated both glucocorticoids in a dose-dependent manner. There was a sex difference in both glucocorticoid secretion patterns, and this was also observed in chickens in an earlier study. There was a sex difference in the HLR response to ACTH, but it is not dose dependent. And finally, there was a significant increase in HLR at the ACTH dose that did not increase glucocorticoids. So our results strongly suggest that care must be taken when measuring glucocorticoids or HLR in response to a stressor in terms of time following the stressor, sex, and possibly age of the animal. So there's this lack of consistent results when it comes to transportation stress studies, and it could possibly be due to how long the bird had been stressed relative to when blood was drawn. So we wanted to make sure that we took this into consideration for our study. So possible explanations for our HLR response. ACTH elicits HLR effects directly. ACTH is stimulating a novel hormone or factor that is increasing HLR. Cortisol has physiological effects on the stress response, and cortisol may have effects, but I suspect that corticosterone is the primary glucocorticoid in ducks because there is a very large database that suggests that corticosterone is dominant. So for future studies, I would be interested in looking into the effects of acute versus chronic glucocorticoids on HLR and brain function. And shortly, I will actually be presenting my next talk on the effects of acute stress on Pekin ducks. Another study I'd be interested in doing is map out the distribution of ACTH receptors, because we know in mammals that there are ACTH receptors located beyond the adrenal cortex. Another study I'd be interested in is to explore the differential stimulation and the effects of central ABT versus CRH. And finally, because we saw these sex differences, this strongly suggests that there may also be age differences, because we know in mammalian literature, there are these well-established estrogen receptor, glucocorticoid receptor interactions. So I'd be interested in looking at pre versus post-pubertal ducks, as well as those in reproductive senescence. So because of the sex difference, there's this gonadal component to the stress response, and it's different at different ages. So I would like to thank Dr. Fraley for allowing me to be involved in his lab and getting involved with this research. Thank you to my lab partners and Maple Leaf Farms for supplying the ducks. Thank you to Antec Diagnostics, Finley Research Center, Purdue, Dr. Megan Abraham, and USDA. Thank you for your time. I will be available afterwards to answer any questions that you may have for me about my study.